This is a seminar that I gave at Adelaide University on March 16th of 2026. Unfortunately, at that time, it wasn't able to be recorded, and so I'm recording it again here, and we'll make it available on the SeadragonSearch YouTube channel. What I'd like to go over today is a bit of the history of work done by our team prior to SeadragonSearch, which largely involved genetics in terms of species delimitation, species discovery, and population genetics of the various sea dragons. And so then I would go into SeadragonSearch and the basic outline of the project. And one important story I'd like to tell, though, was the discovery of the ruby sea dragon that dates back to the middle of the 2010s, and then finish up with the SeadragonSearch project and how you could hopefully get involved. So our original Sea Dragon team, in terms of research, was myself and Nerida Wilson, who's now based at the University of Western Australia and also working at CSIRO, and Josephine Stiller, now Assistant Professor at the University of Copenhagen, who did her PhD with me at Scripps in San Diego. We started this project, though, with some initial funding from the National Geographic, with a project that Nerida and Steve Donnellan at the South Australian Museum and I put together. And this began with a dive actually at Rapid Bay in South Australia. And I remember Nerida wondering if anyone had been working on the genetics of sea dragons after we'd just seen a couple. And even though we worked on invertebrates, we got together with Steve, and that was the beginning of our work on sea dragons. And these fish are highly charismatic. They are only found along the Great Southern Reef of Australia. The weedy or common sea dragon is the emblem for Victoria, while the leafy sea dragon is the marine emblem for South Australia. They're certainly of interest in terms of protection and conservation, and as I'll talk about a bit later, the weedy sea dragon has just been listed as vulnerable in the most recent review by the IUCN Red List, and we are currently working on the assessment for the leafy sea dragon. The weedy or common sea dragon, and I'll generally refer to it as a weedy for the rest of this talk, extends from around Perth in Western Australia along the southern coast to just north of Sydney in New South Wales, and then it's also found in Tasmania. The leafy sea dragon, however, has a more restricted distribution and is known from Western Australia and South Australia only, with some older records potentially putting it into Victoria, but it hasn't been seen there for years. One of the first questions that we wanted to address was the possibility that the weedy sea dragon and the leafy sea dragon respectively actually represented two species, because there were available species names for Western and Eastern weedy sea dragons and Western and Eastern, or Western and South Australian leafy sea dragons. So Nerida and I did an initial paper, first of all, establishing the relationships of the sea dragons, which turned out to be most closely related to a group of Solignathan pipefish within the Cygnathidae, and they were each other's relatives. And we also explored this unusual fish called the ribbon sea dragon that's known from northwestern Australia, and as you can see from the phylogenetic tree here, it's actually not closely related to the other sea dragons, and it's really showing a form of convergent evolution. The sampling we did for each of the sea dragons was a Western and South Australian leafy sea dragon, and Western, South Australian and Victorian weedy sea dragons. And as you can see, they, based on the branch lengths here, they were really genetically very similar for each of the respective species. So we were also able to conclude that there was really only one species of each. This then led to our more detailed work on the genetics within each of these two species. So we recruited a student, Josephine Stiller, to work on the population genetics of sea dragons. And we started off using just what was current at the time, which was Sanger sequencing of some mitochondrial genes. These are often called barcoding genes, like cytochrome oxidase subunit 1. And as you'll see, we actually evolved with the evolving sequencing technology over the following decade. But initially, Josephine was working on this CO1 barcoding gene, and that was what we would routinely sequence first for any fish that we sampled. And we sampled several hundred fish of each, many of them live, caught while scuba diving, and we were allowed to take a small tissue sample. But we also were obtaining museum-based samples if the fish had been fixed and preserved in ethanol, which is actually fairly unusual. But we did get a sample from the Western Australia Museum that Josephine did sequence, and this was not fitting in with the two species we knew. This particular gene, if you looked at it, comparing a leafy sea dragon with a weedy sea dragon, they were about 12% different. So that means for every 100 base pairs of the gene, they are varied by about 12. But this particular sequence varied by about 7% from a weedy sea dragon and by about 13% from the leafy sea dragon. So that 7% different and 13% different meant it wasn't really either of these two species. So we then wrote to the Western Australia Museum and asked if we could borrow the fish, because all we had was the tip of the tail. And they kindly sent us along the fish, as well as a picture of the fish, which actually showed that it was a male with a brood. This had been very fortunately photographed after it had been trawled off Western Australia in the Research Archipelago. And we immediately noticed that, well, one, it had no appendages. There were where the appendages would normally attach in a weedy sea dragon, and it had been catalogued as a weedy sea dragon into the Western Australian Museum collection. But its snout appeared unusually long, and it appeared to be muscular or more robust in the pectoral region here, where the swimming fins are. And also, when we counted, it appeared to have an extra vertebra compared to a weedy sea dragon. So the fish was borrowed, and Josephine and I arranged for it to have a CT scan at a medical facility. So because they're so bony, it showed up beautifully in the CT scan, and here it is in false color. And some of the morphological details you can see here in this plate on the right. And this was written up as a paper led by Josephine along with Nerida and myself, published in Royal Society Open Science in 2015. And the genetics established it as a distinct species. We sequenced both eggs from the brood and from the male and compared those to the weedy sea dragon and the leafy sea dragon, and it was distinctly a different species and was closest relative to the weedy sea dragon, with then the leafy sea dragon the most distant. The species we named in the genus, that is the genus for the weedy sea dragon, so that's Philopteryx, and we named it after Dewey Sea. Dewey White here shown with a picture, a paper mache model of the dragon. And this was because Dewey funded our research. After our initial National Geographic grant, Dewey, based in America, who was very fond of sea dragons, funded the project that allowed Josephine to do her PhD with us and to fund all of the genetic work that we did. And without her, we would not have found the ruby sea dragon. As well as the fish that was originally trawled off Esperance into the west, to the east of Esperance, in a place called the Research Archipelago at about 51 meters in 2007. Nerida went through some collections and asked colleagues in Australia to look, and based on the anatomy, we were able to now tell museum specimens even if we couldn't sequence them. And these fish here, you can see, were trawled off 72 meters off Perth in 1955, and another specimen had been stranded at Cottesloe Beach off Perth in 1919. So the ruby sea dragon had been around. It just hadn't been recognised. And once we did publish the original observations, the species discovery, it got a bit of media attention, and Zoe Della Vedova found one walking further east of the Reshet's archipelago with her kids, and took this photo of what is clearly a ruby sea dragon on a rock, and kindly allowed us to publish that photograph. And other records then started to appear, with another fish washing up west of Esperance and another one washing up south of Perth in Busselton. And this led us to start to wonder if it would be possible to go and find the ruby sea dragon alive in the wild. And so I had a talk to Dewey, and we were going to be going to Western Australia to do some more genetic sampling of sea dragons for our population genetics project in April of 2016. And we added on then, thanks to Dewey, an expedition where we would go to the Reshet's archipelago from Esperance, and we were able to charter a small fishing boat, and the goal was to go from Esperance over to the Reshet's archipelago, and we actually had the exact localities in the two red dots you can see there, where the trawl net that caught the fish back in 2007 had been deployed and recovered. We decided not to do any scuba diving because of the depth and logistical challenges involved with that, but instead to take with us a small remote operated vehicle and to get imagery of the sea dragon. We weren't intending to collect one, and we took along a skilled pilot as well. And Daff is shown here, and here's Col, our skipper, along with Josephine. We arrived on April the 4th after about a day transit from Esperance. Unfortunately, on April the 5th and 6th, the weather was pretty rough. When we put the ROV down to the bottom at 50 odd meters, all we saw was sand. It was very turbulent down there, so the swell was reaching down over 50 meters. But on the final day, before we had to leave, we got lucky, and the swell had dropped, and we were able to deploy the ROV. And so here's Daff doing the piloting, and here's Rusty, who was our photographer from the Western Australian Museum, and Josephine and Nerida there are watching this feed as Daff pilots along. And what I'm about to show you next is actually when we moved indoors to a cabin. What you can see here is a GoPro feed that Rusty set up with Cole and Nerida and Josephine watching the feed while Daff is doing the piloting. And here Daff is now going to follow the sea dragon, and we were able to follow two of the sea dragons. So a moment of great excitement, and we'd just about given up hope, and it was really one of those wonderful moments in science. And this is the video. It's at 54 meters down. It's still pretty turbulent down there, but the swell had dropped enough for us to get good imagery, and the visibility was remarkable because this is ambient light. So the water is very, very clear to be reaching down there. Of course, there's very little red light at this depth, so the fish, which we know are bright red, are showing up as a fairly dull red here. And a lot of the things that are showing up quite dark, like the sponges potentially, were also relatively red. And the fish was clearly interested in backing up against these sponges as we followed along, and seemed to be using them for camouflage. But it wasn't too perturbed by us, and we often saw them doing feeding strikes over the two fish that we watched over about the 40 minutes of the dive that we followed along. And there you could see a feeding strike. They appear to look up and get a vision based on the silhouette of their potential prey. And this initial sea dragon we followed for about 25 minutes, and it actually led us to a second sea dragon. And the two sea dragons encountered each other briefly. Here, there's the second one is there just behind that gorgonian or sponge. And they did see each other, but they then went off in separate directions, and we decided to stop following the first sea dragon, and then we followed the second sea dragon. And The second sea dragon and the first both are black appendages, and both of them appear to have a prehensile tail, which is not present in the other sea dragons, by the way. And we didn't see it holding on, but we think when it was very turbulent and surgy, they probably would have held on to save being really swept away. We, in the end, made this video that was a supplemental document, that an image and video that goes with this paper that was also published open access in 2017 on this journal, Marine Biodiversity Records. Unfortunately, that journal, the link to the online video is no longer available. So if you want to watch it, you should go to my personal YouTube page, and you can then see the video there. And we'll make it available also through the SeadragonSearch web page as well for YouTube. So if you're interested in reading more about the ruby sea dragon, there are the two papers that have been published to date. But in terms of our real goal, in terms of the genetics, we published a series of papers that involved the various kinds of DNA sequencing. And the first one was led by Nerida, and in this case we did DNA sequencing of the mitochondrial region and just a couple of genes. And you can see the circles represent individual genes, sequences, and the larger the circle, the more individuals shared that sequence. So here in Eastern Australia, we showed very little genetic diversity for the dragons from Tasmania up to Sydney. But as you went to the west, there was relatively close relationships to the eastern dragons in Victoria and South Australia, and then a bit of a break indicated by all of these dots here to Western Australia. But overall, the genetic diversity was clearly still supporting the fact that they were a single species, and that within South Australia we probably had the highest genetic diversity, and that there was isolation of Western Australia from those populations more to the east. We followed this up with another paper then led by Josephine in 2017, and here we used, in addition to mitochondrial DNA fragments, we used microsatellite markers from the nucleus. And what we saw on the bottom was the mitochondrial diversity, and there was very little mitochondrial diversity for the sea dragons from South Australia here over to Western Australia. And with the microsatellites, we saw very clear evidence of isolation from South Australia and Western Australia, with little evidence of any gene flow between the two regions. And there was greater genetic diversity in South Australia than we were seeing in Western Australia. So those two papers came out, but about that time there was developments of next generation sequencing technology that we took advantage of using our original DNA extractions of those hundreds of fish that we had sampled. And here we were able to use what are called ultra-conserved elements. And this resulted in two more papers, and the first, led by Josephine in 2021, concerned the leafy sea dragon. And what we had was these ultra-conserved elements are usually used for phylogenetic trees. Because they're conserved, they can be used for sequencing across relatively deep phylogenetic relationships. And in fact, we used it for an overall evolutionary tree of all of the cygnathids, the pipefish and seahorses and sea dragons. But also, if you did sequencing out from these ultra-conserved regions, which is possible, using Illumina sequencing, we were able to then find more variable regions, and this allowed Josephine to mine out what are called SNPs, single nucleotide polymorphisms, and this allowed some wonderfully nuanced inferences compared to what we had done with the mitochondria and microsatellite markers. And the takeaway message from this was that, again, we did see a distinct break between South Australia, the various ways of analysis you can see here showing variation in South Australia was much more marked compared to the tight clustering that we could see for Western Australia. And there was a distinct break between Western and South Australia, and the South Australians showed much greater genetic diversity, as we had shown previously, than what we were seeing in Western Australia. And you can read about this paper, especially in relation to some fantastic work that Josephine did on tracking sea level change and dating back the sea dragon populations to when the Spencer Gulf and the Gulf St. Vincent were both dry land. And they must have then colonized again when these gulfs were flooded again after the last glacial maximum about 10,000 years ago. In this kind of plot, you can see the genetic diversity in South Australia is much higher than the genetic diversity in Western Australia. And this relates to potentially the availability of their shallow water seagrass and kelp habitats that would have been much reduced at the last glacial maximum compared to today, when there's actually much more shallow water habitat for the sea dragons than there was previously. We did a similar study in 2023 on the much richer data set that we had for the weedy sea dragons, since they extend all the way from Western Australia to South Australia, then to Victoria and New South Wales and also Tasmania. We did see markedly lower genetic diversity in the east, in Victoria, Tasmania and New South Wales here in the purples and browns, compared to the South Australian and Eastern Australian Victorian populations here, clustering here, compared to the very low genetic diversity that we were seeing in Western Australia. These UCEs were useful even to the point where, one, the data would allow us to tell siblings, but also even we were able to identify the populations where the region where two poached sea dragon individuals had been taken from, which happened to be in Western Australia. But the genetic diversity, the takeaway message was the genetic diversity in South and Victoria, South Australia and Victoria, was much greater than in the other regions. And we were also able to infer that the colonisation of Bass Strait and the Port Phillip and Western Port Bay shown here, these were dry regions for much of the last 2 million years. The sea dragons came and colonised the Melbourne region from the west and not from the east, even though there's a small signal of genetic connectivity up into New South Wales from Victoria, but mostly the signal is that they came over when the Bass Strait was flooded after the last 10,000 year glacial maximum. They came from this region. Josephine also did a molecular clock, and we were able to date the divergence within the weedy sea dragon populations going back a bit over 500,000 years, with the South Australian and Victorian populations sharing ancestry with Eastern Australia, and then the isolation of Western Australia is shown markedly here. The ruby sea dragon, as I showed before, is most closely related to the weedy sea dragon and separated about 4 million years ago. And then the origin of sea dragons goes back around 7 million, could be younger, could be older, based on the grey bar, and the leafy sea dragon's the most close relative to the other two. So we now know that the ruby sea dragon is a Western Australian species. The leafy sea dragon goes along from Western Australia to South Australia, and the weedy sea dragon has a much greater range. We're continuing to do whole genome level sequencing now of the leafy and common sea dragons, based on the fact that their whole genomes have been sequenced to a chromosome level, and that then allows a much even more detailed population genetic studies than we did with the UCEs, and that's being led by Josephine Stiller and continues to this moment. You hopefully can read about that work in the next couple of years. So, from that original work on genetics, in the late 2010s, Nerida started to wonder if the images of sea dragons, the photographs, could be used to identify individuals. And this proved to be true and allowed us to then launch SeadragonSearch in October of 2020. And the goal is to run a long-term data collection project across the entire range of the sea dragons. And this is also thanks to Dewey White for funding our work to continue on after the genetics and to now look at a much more of a snapshot view based on the participation by the public in providing us with images that they upload through our online platform, and we can then use artificial intelligence to identify the sea dragons. It's a pretty simple submission platform with a drag and drop provision of the images, and then a simple reporting form stating where you got the sea dragon from and your contact information. And then it's after the initial AI processing, a researcher then goes through and confirms the identity of the sea dragon as a new individual or a dragon that has been previously matched. And we're hoping to and have been able to build time series for individual sea dragons. It's proved to be resulting in some very exciting research outcomes. Our team is led by Nerida Wilson at the University of Western Australia. Myself, Chrissy Tustenson has just completed her PhD, the first PhD resulting from SeadragonSearch, and Keisha Ware over here is our first master's student out of SeadragonSearch as well. Jason Holmberg at WildMe, based in the US, is the lead scientific software developer for the project. He's at Conservation X Labs, and without the cooperation of WildMe, SeadragonSearch would be nowhere, so we're very grateful to Jason. We're also grateful to Dewey White. Our other team members are Bronwyn Jalanders, our South Australian representative, Cade Mills, our Victorian representative, Keith Martin Smith, our Tasmanian representative, Dave Booth, our New South Wales representative, and Anka Kluter, based here in California, and Erin van der Ven, who did a wonderful Masters of Advanced Studies capstone project interviewing some of our wonderful contributors to SeadragonSearch, and you can see those also on our YouTube channel. A little bit about WildMe if you want to go and read about them, and there are various kinds of projects involved in using images to identify all sorts of different animals for conservation purposes. Once again, we thank WildMe for their collaboration over the course of this project today. What's great is that after the initial training of the algorithm to allow the WildMe platform to identify sea dragons in photographs, it is then able to use subsequent algorithms that they've developed to identify dragons based on spot patterns or missing appendages or other scarring or damages. And this happens for both the leafy and the weedy sea dragon. And the way that it works is after the photograph is uploaded by a person, the image is then, the dragon is found in the image, and you can see it doesn't have to be portrait quality images. Often they're even GoPro images that are submitted by people. We can use those, and the image is then processed through, and we get a ranked series of matches based on this number out of a ranking of up to 1. And here you can see the top 3 rankings are all for the same fish, WA2153. And then the 4th ranked fish is WA1578, and the numbers go down. All of the matches are from Western Australia, but there's actually 1 match from New South Wales. The images then are checked for the top ranked ones just to see if that really is a good match. And you can look and see that, oh, this flag here has a little white tip on it. The flag there, it's not a great match on that particular photograph. Do the spots match up? And so someone does go through and do a check to see if it is the same fish. And then it is, once it's confirmed, and in this case, this match was seen to be a better one than this top one here. But it was definitely that fish. And so if it's a new fish, it gets assigned a new individual name. If it's a spotted fish that's done before, it is then assigned that individual number from the previously identified fish. So after the identification is performed, the submitter will then get an email, and will also get subsequent emails when that fish is seen again. You can go to our seadragonsearch.org platform. At every page there is a link to submit. There is also a training video on how to submit under our What is SeadragonSearch drop-down, and you can submit your photos there on a button, as well as watching these YouTube videos on how to actually do the process. Because you are taken out then to the WildMe site from our SeadragonSearch site. We also offer a SeadragonSearch iNaturalist project page where you can also submit to us. It's just this doesn't go directly to SeadragonSearch, and our team has to go in and pull these images out and then submit them on your behalf to our WildMe platform. So we'd prefer if people did submit directly to SeadragonSearch, but we're perfectly happy to take them from iNaturalist as well. Do note that we are SeadragonSearch for our iNaturalist project, not DragonSearch South Australia, which has a separate iNaturalist project. How has it been going? Well, as of February of last year, 2025, you can see we've had over 7,000 images submitted from Western Australia, 5,000 from New South Wales, over 3,000 from Tasmania, and over 4,000 from Victoria. We are saddened that our engagement in South Australia has not been that great, especially since I pointed out the great genetic diversity of sea dragons, both species, in South Australia. So we would really like to see more images submitted from South Australia. As of March this year, just last week, we actually hit 24,000 images. And we regularly provide yearly, certainly a summary of the progress through our Instagram and Facebook pages. And this was our report card for 2024. In 2024, by the end of 2024, we were up to nearly 19,000 images, up from nearly 14,000 in 2023. In terms of the number of individuals, we were at nearly 5,000 at the end of 24, up from just over 4,000 in 2023, with leafy sea dragons over 550, up from 4,436. We spot things like this, such as the earliest brood in each year was Hervé from New South Wales on June of 2024. The most sighted sea dragon was called Nicole, 74 encounters just from New South Wales. And the most encounters in one day was in August of 2024, where there were 60. Every month the leaderboard is updated through our Instagram page. You can see that. We have an annual First Broods competition. We have a sponsoring Sea Dragon program, and we regularly have in-person meetups at the various sites in Australia. Outputs include podcasts, contributor spotlights, which can be videos, public presentations, and a documentary trailer in 2024. In 2025, we reached that over 23,000 submissions, over 5,700 weedy sea dragons, and over 600 leafy sea dragons. The earliest brood was in Western Australia in 2025. Lucia travelled over 3 kilometres from Cottesloe to Perth to Port Beach in Western Australia in 2025, and our longest time between sightings was 4.5 years at Portsea in Victoria. All of the usual engagement continued with an increased number of followers on Instagram and Facebook. We redesigned our website in 2025, and we had 2 graduate student completions in 2025, Keisha and Chrissy. In terms of South Australia, which I decided to talk a little bit about because I was giving the talk in Adelaide, in 2025, this sea dragon, a leafy sea dragon, was seen at Rapid Bay, SA 0275. It was seen in April of 2025, again in July of 2025, and again in October of 25. And here it was apparent that it was a male with a brood on his tail. This individual was seen when the harmful algal bloom impact in the southern Australian Gulfs began to impact. It has not been seen, though, unfortunately, after October of 2025. This fish, SA 0280, was seen at Tumby Bay in South Australia. The harmful algal bloom hit Tumby Bay in late May of 2025. The fish was seen in 2025 in June and was not seen after that time. I was fortunate enough to go for some dives in Rapid Bay in middle of March and did see one at the Rapid Bay jetty. I did submit the images that I took to SeadragonSearch and then received this email because it was quickly processed by Nerida that said this fish actually had been previously sighted. And so I was then told a little bit about that, and this was the fish. Jemima was photographed the fish in January of 2026, and then my image was taken a few months later at the also at Rapid Bay Jetty, and it's assigned the individual name SA293. Notice Jemima's fish was taken without a flash. I was taking with with a flash, but I took very few pictures of the dragon. And this was enabled by Peter and Ilona Corrigan at Seadragon Dive Lodge at Second Valley, and thanks to Carl Jarder for guiding me around and helping me find that sea dragon. This is the only sea dragon we've had lodged on SeadragonSearch so far in 2026, so I encourage all South Australians or other visitors to South Australia to please contribute. We'd love to have your images. Note, we have a code of conduct in terms of trying not to stress sea dragons, certainly not handling sea dragons. Good photos, not great photos, are not needed. You can avoid using a flash. As you can see, Jemima took a perfectly great image, especially hitting them with hot lights. Remember, these fish don't have eyelids and they do get stressed if you hit them with too much light. So please read our code of conduct if you are going to submit to our program. I'll tell you a bit about our outcomes in terms of abundances, reproduction, the reassessment for the red list, and the upcoming reassessment for the leafy sea dragon. And this was all based on Chrissy's PhD work. She graduated last year, and one of her thesis chapters is now in review. It's come back from review, and we're revising it for resubmission to Fish Biology and Fisheries, which gives us some abundance indications. Chrissy also led the IUCN Red List reassessment of the weedy sea dragon that was published last year, and she has also led a paper on the conservation of sea dragons and the overview of what's known about sea dragons' future directions, and that was published in Fish Biology and Fisheries last year. Our trends paper is in revision for marine biology, and Chrissy is preparing a breeding paper based on their observations as also another thesis chapter. So here's the IUCN reassessment that you can see online at their website. Now they're listed as vulnerable. And here's the paper in public open access in Fish Biology and Fisheries reviews. In terms of sea dragon abundances and trends, it really is needed that some subsequent work should be done based on the assessments that were done now up to 2 decades ago, especially following this paper by Edgar et al. That was published in Nature for the Great Southern Reef, and noted that there had been a marked population decline for the weedy sea dragon, although no sites were really indicated in that study. And for IUCN assessments and management, abundance data is really needed. So what Chrissy did was to go through and look at sea dragons based on their imagery that had been submitted to the project. And because we had so many images from New South Wales, Victoria and Tasmania, it was decided to restrict the project to the paper to that region. And here you can see the overall region of Sydney, Melbourne and Hobart. And she really focused on Cornell, where we had many images submitted, but also the Portsea Pier and Flinders Pier in Melbourne, and then off Hobart. And what Chrissy was able to do at these sites was to use theory based on the amount of photographs that were taken, and that as you continue to take photographs in a particular region, you should start to see a plateauing in the number of fishes as you keep photographing individuals and resighting them. And this fits into mark recapture methodology, and what we were able to see with the data is that there were plateauing of the sea dragons as the number of encounters increased. And at Cornell in Sydney, there were nearly 4,000 encounters, and the numbers were plateauing at about 300, though still slightly increasing. But you can see that at the 2 Melbourne sites, at Flinders and Portsea, the curves were still not plateauing at 400 or more individuals for Portsea, and well over 800 individuals for the Flinders region. For Tasmania in Bicheno, just north of Hobart, it really did start to plateau. with about 200 individuals. And this mark recapture theory then allows for assessments of the population size overall and whether it might be tending to increase or decrease. And there are assumptions in the models that are being used, which we've addressed hopefully in our revision, and the sampling effort is inconsistent and all based on the submissions based on citizens, so members of the community. So there are weaknesses in this method, but we hope that the signals that are coming through based on the thousands of images outweigh the noise that we're getting. The basic takeaway message is that at Cornell and at Flinders and at Portsea, the numbers are increasing or stable, while there's definitely a decreasing trend for Bichonneau. And this played into the IUCN assessment of the weedy sea dragon. Certainly, we need to keep monitoring what's going on at Cornell as well. But certainly in Victoria, there's a healthy population of each at each of the two sites that were monitored. What's notable about Tasmania, it's a climate change hotspot with warming oceans down there and decline in kelp habitat. So we really do need to keep an eye on the Bichonneau and Hobart regions of Tasmania. In terms of reproduction, there's been studies done on brooding occurrences of sea dragons, and there's been work done on the development of sea dragons after they've hatched. But there's very little information available for leafy sea dragons at all. And apart from the regional observations just for New South Wales, we needed to do a more range-wide survey for the sea dragons. So Chrissy went in and looked at the occurrences of broods on males. And this was able to importantly track the first observations of broods in a particular year. Also when broods stopped being seen. Information based on the number of photos that were taken allowed us to interpret the length of a brood. How long did a male carry the eggs? How long or how many times was a male able to have broods? And so this is work that's in preparation. It has yet to be submitted for peer review, but the basic message is that in Western Australia, which are at similar latitudes, where the water is warmer in winter, they were breeding first. So these were breeding in June and July, whereas in southern Western Australia and in Victoria and Tasmania, they might not breed until much, much later on. And then they would also tend to breed later on than what we were seeing in Western Australia and in New South Wales. Chrissy was also able to see the number of broods that range from 1 to 3 in the different regions, or 1 to 2 in the south. So in warmer waters, they could breed more often in a breeding season. And the incubation period was longer in the more southern, colder regions than in the warmer regions. And the interval between broods was kind of unclear. It was no clear signal in that. Algae is something we often see on broods, and this varied according to site as well. So, recruits, we were also able to look at the onset of breeding, as I've already spoken about, but also when did we start to see the little juvenile sea dragons, and that also varied with earliest brood recruits being seen in New South Wales and Western Australia off Perth, and them showing up later on in the more colder southern regions. So look for that paper hopefully coming out in 2026, ideally. The weedy sea dragon story in terms of its red list assessment was most recently in 2017 that it was of least concern. Based on the work that had been coming out of SeadragonSearch and the paper by Edgar of a decline overall and issues like population affected potentially by global ocean warming, the assessment changed the weedy sea dragon to vulnerable from least concern. And we are now conducting the review of the leafy sea dragon, and that should be coming out in 2026. If you're interested in SeadragonSearch, we have platforms on Instagram and Facebook, regular posting where you can see what's going on. When the first broods happen, there's a contest. If you want to participate, you submit your images, and if you have sighted the first sea dragon brood in your particular state, you'll get a cup. We monthly have a leaderboard of the contributors submitters and photographers who've contributed most each month, and there's a lot of competition involved in that. And we also point out interesting re-sightings or novel sightings, and so there's lots of things to look into there as SeadragonSearch continues. And I'd just like to thank all of our partners and team that all make this a very successful project so far, and we'd really encourage more submissions. And South Australia, we'd love to have more information coming in from you. And thank you very much for your attention.